everyone and welcome to this video. In an earlier molecular docking video, I showed an end-to-end in silico pipeline to derive a PDB of a protein ligand complex. The example I had used was the docking of amyloid beta plaques with the monoclonal antibody lecanemab in context of Alzheimer's disease. However, for a drug to be viable, such a complex must be stable in vivo environments. These environments could be blood, saliva, or cerebrospinal fluid, based on where the drug complex has to perform its function inside the body. Similarly, biomolecules used for drug delivery, for example liposomes or protein-based nanocarriers, should have specific behavior such as triggered delivery or potential for cellular uptake through membranes, like the blood-brain barrier. In silico behavior modeling using molecular dynamics tools can be extremely valuable before moving to more resource-intensive in vitro or in vivo studies. It reduces failure rates and accelerates discovery. In this video, I will show such a simulation using GROMACS, a molecular dynamics tool. I will simulate factor XA, a protein playing a critical role in the formation of blood clots. As before, the 3D structure is available from the PDB as code 1FJS. We will model the behavior of this protein in a simple aqueous system. So let's go through the notebook step by step. We first prepare the imports and obtain the input PDB file. Then we do a bit of cleanup to strip out atoms that do not belong on the protein. We then generate the topology for the molecule and some other necessary files like position restraints. This will have all the information necessary to define the molecule within a simulation. The next several steps are essentially to review the generated files and understand them. So we will just run through these cells quickly. The next big important step is solvating the system. Here, as I mentioned earlier, we use a simple aqueous solution with some ion. Essentially, we define a box and place the protein molecule properly so that spurious interactions with periodic images of the molecule are not introduced. We then fill the box with water molecules uniformly and visualize it. We then need to add some ions because we have a charged protein which must be neutralized to model approximate physiological conditions with a NaCl concentration of 0.15 molar. Here, sodium ions are purple and chloride ions are green. Before we can begin dynamics, we must ensure that the system has no steric clashes or inappropriate geometry. The structure is relaxed through a process called energy minimization. To see if the run worked, the potential energy should be negative and of a certain value, and the maximum force, Fmax, should be below a target. The resulting plot should show a steady convergence of the potential energy as seen here. We use position restraints to relax our solvent and ions around the protein without letting the protein move. Next, we equilibrate the system in two phases. First is to get the system to a simulated temperature by modifying atom velocities according to the predicted kinetic energy distribution. This is a time-consuming step, so we use previously computed results. This shows the temperature of the system reaching the target value of 300 Kelvin and remaining stable. Next, we must stabilize the pressure and density of the system. Again, we use a pre-computed run here and observe the results. There is a fair bit of fluctuation, but the average pressure remains on the same order of magnitude as the target. And if we see the density, the average stabilizes a bit higher than that of pure water due to that protein. After the system is at equilibrium at the desired temperature and pressure, we are now ready to release the position restraints of the protein atoms and launch our production MD simulation for data collection. Again, the run takes time, so we use pre-computed output and analyze it. We will focus on a few important pieces of analysis. First, we can visualize an animation or the trajectory, which intuitively shows structural stability. We can then evaluate structural stability with root mean square deviation in the solvent relative to the original crystal structure. And we see that it is small and stable around 0.15 nanometers. And we can look at compactness of the structure with its radius of gyration, which remains very stable in its compact form. In future work, I plan to model the composition of the cerebrospinal fluid in the solvent box and the lecanemab amyloid beta complex as the molecule and run the MD simulation to show structural stability in vivo. I hope you found this useful. Thank you for your time.